awesome researcher that's been working with Angelman Syndrome Foundation and in the Angelman Syndrome space for so many years for our families, Ben Philpott, and they're going to give us a research update. So this is a really exciting time for this community, and I think you're going to hear it in this update. So I'm excited that we're able to bring this to you guys, as the last couple of days, all these individuals, as well as about 100 other individuals, have taken part of the research symposium, where they're working really, really hard for our families. So they're going to give you an update, so I hope you enjoy it.
Thank you, Amanda, and thank you all for coming. I know how hard, I can't imagine how hard it is for you to make the voyage out here with your families. I'm in awe, so it's really a pleasure to be here for you. So over the past day and a half, as Amanda said, we've been holed up in the room next door, around the corner, talking about research. And how this works is, in general, Ben and I have chosen numbers of people who we thought we would like to hear talk at the research conference, but also scientists submit their new findings to us, and we give them an avenue to present. And so the updates that we're going to give today are both things that have happened in the literature, news that has come out since the last conference 2 years ago, as well as new work that we heard about next door. Okay? So to give you an outline, research is big, and there's a lot of different moving parts to it. We will first talk about basic and translational research, biochemistry, mouse studies. These are the things that are hard to understand and probably won't impact your families immediately as much, but nonetheless, they're very important things to do, and we'll get into that. The next set of topics are going to be the therapeutic approaches. There are some very exciting things going on for Angelman syndrome in this area, and we'll give you an update about that. And finally, we're going to talk a little bit about clinical studies, because this is going to be important to move the therapeutic approaches to you guys. And so we'll talk about what's known there and what ASF is doing to make sure that we can translate the research to therapeutics for you guys. So first, we're going to talk a little bit about what does UBE3A do. UBE3A, its fancy name is an E3 ubiquitin ligase. You don't need to know that. What you really need to know about UBE3A is that it puts little tags onto proteins, and that little tag signals for that protein to be thrown in the trash, okay? And so when your kids are missing UBE3A, there's a buildup of trash. And we don't know what that trash is, and that's kind of tough. But nonetheless, that's what's happening, is the UBE3A puts a signal to send a protein to the trash can. So what proteins are targeted? Like I said, we don't know very much, but there's been some insight over the past couple of years. First of all, proteasomal subunits. And so this is the trash can. UBE3A actually interacts with the lid of the trash can. And we don't know what that means yet, but it's important to know that, because at least we kind of know one of the things it's stuck to, okay? It's stuck to the trash can, and that's important to understand. There was another recent paper about a protein phosphatase 2 phosphatase activator, and I don't know what that is either. But importantly, we know that this may impact neuron function, and so we watch these things carefully. And what's cool is in science, an important part of the scientific process is to see things repeated and reproduced in other labs. And so we anxiously await things to be repeated, or many of us will try to repeat these things in our labs to make sure we believe it and to make sure that it's solid work. New information about UBE3A protein that's come out in the past couple of years. One important feature is we're learning more about the structure of UBE3A. So I told you it's a protein that tags other proteins to go to the trash can, but it has different pieces itself, and the different pieces are important to understand so that we can know how it functions. For instance, this little yellow box here called the hex domain, that's the part that it puts the tag on. It's the business end of the protein. But the protein requires these other pieces, like the azul domain, in order to do that efficiently and to name its targets, perhaps. And so in the last year or so, the understanding of this azul domain has become very important, and there's been 2 major papers out on the azul domain. And ultimately, another reason we need to understand this protein structure is so we can interpret how which mutations cause UBE3A, or cause Angelman syndrome, and where they're located and how they might do so. How do mutations in UBE3A lead to the loss of function? The next question is where in the cell is UBE3A? This is a somewhat important question because where in the cell helps to determine what its real function is. So I told you it's tagging proteins and sending them to the trash, but we don't know which proteins. And so if we knew where it was located, we can take a better guess at which proteins. So this little ball in the middle of the cell is the nucleus. It's where the DNA is. UBE3A may play an important role in the nucleus here, and so if it's near the DNA and doing something in this space, that also might be important. But if it's out here in the cytoplasm, it can do other things. And for instance, all of the little neuron arms that are reaching and touching other neurons are considered cytoplasm. And so UBE3A out there might be doing something specific in a neuron. So there are 3 different forms of UBE3A. They're descriptively labeled as 2 long ones and 1 short one, or 1, 2, and 3, which is even more descriptive, right? But the 2 long isoforms of UBE3A were recently shown to be found in the cytoplasm, and the short isoform is found in the nucleus. And some ASF-funded research from Ben Philpott's lab showed in human neurons where UBE3A is localized. It says that it's in the nucleus. And we heard this exciting talk at the Scientific Symposium reporting on a new recent publication from Ipe Gersma's lab. He's at Erasmus University in Rotterdam in the Netherlands. And he was able to show that when you get rid of the short isoform, or the one that's found in the nucleus, that alone causes most of the symptoms of Angelman syndrome in the mouse model. And that tells us that short isoform, that one that's found in the nucleus, is very important for UBE3A function and for Angelman syndrome. And ASF funded this research as well, and so we're very excited to hear that study. Now I'll turn it over to Ben, who will take over.
We're going to attempt to go back and forth a number of times on this. So as part of the research efforts through the Angelman Syndrome Foundation, a very important aspect is to perform studies in mice that could eventually lead to new treatments. So there have been a number of mice that have been genetically engineered that provide powerful tools that allow us to identify the roles of UBE3A, the times in which it needs to be expressed, and to test possible therapeutics. So for example, we've designed mice in which we can turn on UBE3A at different times during development and even in different places of the brain. This is really important because it will tell you, for approaches where we're trying to reinstate normal UBE3A expression, how early do we need to do that? Do we have to do it in newborn individuals, or could we do it even in adulthood? We're also looking at studies where we have mice where we can remove UBE3A at any age we want. And this is important because it tells us how long we would need to maintain UBE3A expression. Do we need to maintain it throughout life, or just during developmental periods? We've also engineered mice that can overexpress UBE3A. And this is important because it will tell us in approaches where we're delivering UBE3A, such as in a gene therapy, it tells us what's the tolerance of being able to have overexpression without having any adverse consequences. So it's giving us some therapeutic guidelines. So the role of all these studies is to define treatment parameters for when we eventually go to the clinic. And we want to do these studies first in mice so that we can empower eventual clinical studies. So we're answering questions such as, what's the critical window for expressing UBE3A? When during development does it need to be expressed? What brain regions do we need to deliver UBE3A to? Are there any particular neurons that we need to get UBE3A expressed in? And what are the consequences of having too much UBE3A expression? So there are a number of new findings that have come out just in the last year, and we want to give you a little flavor of some of those new findings. So for example, work from my lab showed that if we reinstate UBE3A expression, sorry, I'm on the wrong slide. If we reinstate UBE3A expression, that even if we do this in juvenile mice, this can improve epilepsy outcomes. So this is very important. So it suggests that reinstating UBE3A can have beneficial outcomes for epilepsy. We've also shown that Angelman syndrome model mice have deficits in an area of the brain known as the prefrontal cortex, which is up here. And then work from Ipa Elguerzma's lab has shown that even in adulthood, when you reinstate UBE3A expression, you can restore some of the neurological functions in that area of the brain. Work from Ipa Elguerzma's lab in the Netherlands has also shown that if you delete UBE3A in adulthood, it has very little effect on the behaviors of mice. So this is another clue in saying the most important time of having UBE3A expression would be to have it during developmental periods. So that's a major goal of current research, is to find ways to reinstate UBE3A expression early in life and to maintain it, particularly across developmental periods. Once again, work from Ipa Elguerzma's lab is really providing a foundation for pharmacological studies that are coming on. So Ipa Elguerzma's lab has very carefully characterized behavioral deficits in a mouse model of Angelman syndrome. So this tells us exactly the types of behaviors we can study when we're trying pharmacological or genetic treatments to restore functions in the Angelman syndrome model mice, to get clues as how this might help in eventual clinical trials. So there's also a recent study from Katzum's lab that suggests there might be some differences in behavioral phenotypes between male and female mice with Angelman syndrome, and this is a provocative finding that needs to be followed up on. There's also a new mouse model that studies Angelman syndrome imprinting defects. So the most common models of Angelman syndrome involve deletions of UBE3A. But now there's also a new model that will model the imprinting center defect. So it will allow us to study treatments for that form of Angelman syndrome as well. There are many other models, not just mice, that we need to consider that are becoming increasingly important. So Stormy Chamberlain lab is really famous for getting human cells from individuals with Angelman syndrome. Then these become de-differentiated, so they're pluripotent, and then she differentiates them into neurons. So she has Angelman syndrome patient cells that can be generated into neurons that we can then use to study treatments that are going to humans. Really, if there's a therapeutic approach, such as an antisense oligonucleotide approach, which we'll talk about in a little bit, we need to know that this works in human cells. It's not sufficient if it works in a mouse. It's got to work in a human cell. So this is really the great work that Stormy's lab has been doing. There are additional models as well. So for example, there's a rat model of Angelman syndrome to accompany the mouse model. The rat has some advantages because some of the behaviors are a little bit more robust in rats compared to mice. And now Scott Dindot has developed a pig model of Angelman syndrome. And this provides the first ever large animal model of Angelman syndrome. And I think it could be particularly important for figuring out the pharmacokinetics, for example, of drugs and testing treatment opportunities in a large animal model to accompany those that might be explored in a rodent model.
And so as Ben mentioned, my lab works a lot in iPS cells, and so we thought we would tell you about new findings from iPS cell studies. First of all, my lab has been working on understanding why UBE3A imprinting is restricted to neurons. What does that mean? That means that UBE3A is shut off. Dad's copy of UBE3A is shut off only in neurons. And we want to understand why that happens so that we can understand ways in which to turn it back on. We published that earlier this year. And new other groups have been deriving iPS cells from patients with different genotypes. So for instance, the Steve Haas lab in Germany derived iPS cells from patients with an imprinting center defect. And what's really cool about these cells is we've been, the community's been good about sharing them with the each other and distributing them to other labs and pharmaceutical companies, which helps everyone study the disorder better. So what are the challenges facing basic research? First of all, we need to be cognizant of how we balance investments in basic and translational research with clinical research. The clinical research might have the most direct benefit to you, but we also have to invest in basic and translational research so that we can come up with therapeutic approaches and advanced therapies. And this is how we get innovation and why there's new things that help your kids. Which research study should we be devoting most of our time towards? As you can tell, there's a lot of fantastic mouse studies, IPS studies, protein studies that are really cool, and we haven't even gotten to the clinical studies yet. And it's important to try to prioritize those that are going to be the most help and going to give us the most bang for the buck. But we also have to think about the future, which things are going to lead to the therapies that you guys are going to need in 5 years as well. How well does the therapeutic window translate between mouse and human? A lot of the preclinical studies, a lot of these studies that tell us that things work, happen in mice, but we need to know how they translate to human. And I think for the first time in our scientific meeting, we heard the words non-human primate more frequently than we usually do. And so we also need to understand how do we go from mouse to non-human primate to clinical trials in individuals. And so we need to understand how to use our model organisms. And finally, how do we balance research toward a cure, so the things that put UBE3A back, with research toward development of other therapies to help with Angelman syndrome symptoms. Some of you, you need help now. You need help with the symptomology, and in the future, we don't know what Angelman syndrome is going to look like after some of the therapeutic approaches, and so we still have to be thinking about the symptomatic approach, and that's more difficult. And so we have to remember to do that while we're working on these exciting curative therapies as well. And ultimately, rest assured that we researchers have the ultimate goal of improving the lives of individuals with Angelman syndrome. We do all of this with the thought that we want to help you. And so coming to this meeting and seeing your kids and whatnot remind us that this is why we do what we do. And we encourage through our funding priorities at ASF and through this meeting, we try to encourage researchers to make sure to keep the patient in mind as they carry on about their research.
So one of the approaches that we heard a lot about this meeting and is getting increasing press is using gene therapies to treat Angelman syndrome. And basically what a gene therapy is taking advantage of viruses, and it's taking all the bad stuff out of the viruses and it's packaging in good stuff back to the virus. But you can use this viral package to deliver what you want to the brain. So you can use a couple of different approaches. For example, you can deliver UBE3A to the brain directly, or you can deliver something in a virus that will turn on the silent copy of UBE3A. So if you remember, there are 2 copies of UBE3A, one that's inherited from mom and the other that's inherited from dad. It's the mom's copy that's deleted and the dad's copy is silent. But there are ways that we can activate it and we can deliver that through a viral particle. So the Angelman Syndrome Foundation funded the first ever gene therapy study for Angelman syndrome. This was a study to Ed Weaver's lab, and they've continued to fund gene therapy approaches, including what Steve Gray presented at the most recent conference just yesterday. So there are some important aspects of a gene therapy for Angelman syndrome. One is, as Story mentioned, there are 3 different types of UBE3A, 3 different isoforms we call them. Which is the one that we need to deliver to get maximal benefits? And this is why that work from Ibel Gersma is so important, because he showed that this short isoform of UBE3A is really the one that's important for regulating Angelman syndrome phenotypes. And now we know which one needs to be packaged for gene therapy. So what are the expectations for recovery based on age of intervention? So work, once again, from Ibel Gersma's lab has been carefully mapping out when we reinstate UBE3A expression, what aspects or phenotypes do we expect to recover. For example, you can get some recovery of seizures with early reinstatement. And basically, almost everything can recover if we have early reinstatement. With later reinstatement, we can get some motor improvement still. We can get some improvement in epilepsy. Even in adult reinstatement, you can get recovery for things like synaptic plasticity, which is thought to be the substrate for learning and memory. So that's really encouraging for us. So the idea is that even if there's a later reinstatement of UBE3A expression, we expect it to have some benefit. We need to know whether we need to target specific regions of the brain or the entire brain. How many neurons do we need to reinstate UBE3A expression in? And are there any dangers of having too much UBE3A expression? If so, how do we limit this so that we don't do a gene therapy, which is irreversible, that would cause any undue adverse consequences? So we're trying to be very, very careful and thoughtful in the way in which we're developing these gene therapies. Steve Gray, who is leading the Gene Therapy Center at the University of Texas Southwestern Medical School, is a pioneer in gene therapy. In fact, he developed the first ever intrathecal, which is delivered to the spinal cord, delivery of a gene therapy to treat a rare syndrome known as giant axonal neuropathy. And now he's turned his attention to trying to develop a gene therapy for Angelman syndrome. So he's designed a particular type of viral vector known as AAV9, or I should say he hasn't designed it, he's using it to carry UBE3A and deliver it in a way that mimics the way it naturally occurs in the brain. So in early pilot studies, he's found that he can deliver UBE3A in a widespread manner throughout the brain, and now he's working on long-term safety and efficacy. And this is actually work that he's doing in collaboration with my lab. But it's a very exciting and opportunity. So I mentioned that gene therapy can be used to reinstate UBE3A expression, but there's also this other approach that can be used to reinstate UBE3A expression, and that's turning on that silent copy of UBE3A that's inherited through the father. And now there are no less than 4 different ways of doing this, and this is incredibly exciting because this means we have 4 different shots on goal to develop a treatment for Angelman syndrome. So one approach is to use a small molecule, so just a drug that you can deliver that can unsilence UBE3A. So my lab, in collaboration with Mark Zilka's lab at UNC, showed this as a proof of principle. You can use an FDA-approved drug known as topotecan that can be delivered to turn on paternal UBE3A. Unfortunately, this drug has some toxicities, but now there are others that we are working on that have less toxicities. I'll talk more about this in a second. There's also this wonderful approach known as using antisense oligonucleotides that basically it removes a stop sign for the expression of the father's copy of UBE3A. And this is one that several pharmaceutical companies are advancing. I believe you'll hear from 2 of them today. So that's incredibly exciting. There's also hopefully many people in the audience have heard of this new technology known as CRISPR, which is a new gene editing technology. So Mark Zilka's lab at the University of North Carolina is using a CRISPR-based strategy to downregulate the UBE3A antisense, which is just a fancy way for saying it's removing that stop sign so that you can get expression of the father's copy of UBE3A. And then there's another approach known as an shRNA approach, which Stormy's lab is developing to do the same thing, to remove that stop sign to allow the father's copy of UBE3A to be expressed. And notably, both the CRISPR-based technologies and the shRNA-based technologies can be delivered as a gene therapy through these viral vectors. I think I talked when you were supposed to, but that's okay. So these UBE3 unsilencing approaches have some really important aspects to them. So for example, if you are using a drug to unsilence UBE3A, how long does that unsilencing last? So if it lasts for just a couple of days, it means you have to take a drug every 2 days. If it lasts for a lifetime, that's great. So it means you could get a one-time dose of a drug and it'll last forever. So So one of the exciting things that I'm not going to talk about today is we can give a drug known as topotecan to unsilence UBE3A, and it can unsilence the father's copy of UBE3A for as long as a year, which is as long as we've looked. We also need to figure out what are the most appropriate delivery approaches for whether it's a small molecule or a gene therapy. What's the right way to deliver the drug? Is it something that we deliver orally? Is it something that we deliver intrathecally by injection in the spinal cord area? Or even something we might consider injecting directly in the brain. These are all very important considerations that the scientists and clinicians are working on. We also need to know what level of unsilencing is necessary to lead to clinical benefit. That is, how many neurons in the brain do we have to express UBE3A in order to get clinical benefit? So we're really trying to carefully study this, and we have some hints from actually studies of individuals with UBE3A mosaicism that we might only need to get UBE3A in a small number of neurons to get a very significant clinical benefit. And also, could just a low level of UBE3A unsilencing over a long period of time provide benefit? These are all questions that we're studying at the basic science level. So there have been a number of new findings on unsilencing of UBE3A. So, for example, the Angelman Syndrome Foundation funded work I did in which we've studied now new drugs in a certain class of compounds that is topoisomerase inhibitors that can unsilence paternal UBE3A. So I mentioned that we discovered a number of years ago that an FDA-approved compound known as topotecan can unsilence paternal UBE3A. But if anyone has had the unpleasant experience of watching a loved one go through chemotherapy, you know that a lot of chemotherapeutics have adverse side effects. So now we're trying to study other topoisomerase inhibitors that can unsilence maternal UBE3A that don't have toxic side effects, that have much better safety profiles, that get delivered to the brain very well. And although I'm not going to be talking about it today, we've also identified new classes of compounds that aren't topoisomerases that can produce unsilencing. And that's another really exciting area of future research. At the scientific symposium, we just heard wonderful talks from Noel Germain and Mark Zilka about their approaches to unsilence paternal UBE3A. So Noel is using antisense oligonucleotide approaches and shRNAs to unsilence paternal UBE3A. And she's doing this using the patient-derived cells in Story Chamberlain's lab, and she presented some very exciting results that this can be achieved in patient cells. Mark Zilka from the University of North Carolina talked about his work using CRISPR-based approaches to activate paternal UBE3A. And he also talked about the possibility that this could even be a therapeutic that could be delivered prenatally. So this type of treatment could be delivered throughout life, ranging from being delivered prenatally to being delivered in adulthood. So this presents a whole range of possibilities, and we had a very robust discussion about these types of delivery methods and these treatment opportunities that this could produce. One of the changes in the landscape of Angelman syndrome is the involvement of the pharmaceutical companies. So when I first started coming to Angelman Syndrome Foundation meetings, there was really no representation from pharmaceutical companies. And now there's incredible representation from pharmaceutical companies. And these are real powerhouses and players in the field. So just this meeting, we heard from 3 different pharma companies about their use of antisense oligonucleotide approaches to unsilence maternal UBE3A and potentially treat Angelman syndrome. So this includes work from genetics that Scott Jindot presented data on, and their ASO use and some findings he has in non-human primates. There's also work that was presented from Frank Rigo and Ionis, and from Dr. I might mispronounce her name, Ina Brunig-Travert from Roche, talked about their approach of treating Angelman syndrome using antisense oligonucleotides. So all 3 of these companies have presented very compelling data, and I think at least 2 of them are going to be talking upcoming right after us at this parent conference. Should stress that a lot of the goals are to go for a truly transformative curative treatment, and this would involve reinstating UBE3A expression in one way or another. But we also want to be mindful of the fact that it's important also to treat other symptoms of Angelman syndrome. So be it movement problems or seizures, even if we cannot get a cure immediately, we might be able to quickly help in some of these other phenotypic domains. So this is a large area of research. So in the past year, there have been some studies that have come out that have suggested some incredible opportunities. So for example, there's a study published in which a small neurogenic molecule known as NSI-189 can improve motor and cognitive function in Angelman syndrome model mice. So this is something that's in preclinical development, meaning it's being studied in mouse models with the idea that it could eventually get to the clinic. There's also work from Sylvia Russo's lab showing that an amino acid compound known as taurine could rescue motor and learning deficits in Angelman syndrome. And finally, there was work from Chris Carey's lab, from one of the Angelman syndrome clinics in Mass General Hospital, that buspirone could improve anxiety-related symptoms in individuals with Angelman syndrome. So as you know, anxiety can become an increasing problem, and so it's nice that groups are starting to consider ways to help with anxiety issues. Ovid is using a compound known as OB101 to try to ameliorate some of the phenotypes of Angelman syndrome. So one of the discoveries that actually came from Angelman Syndrome Foundation funding of Dr. Agawa's work is that there's reduced tonic inhibition in the brains of Angelman syndrome model mice. But they really didn't know what the functional importance of that was. Ovid is using a compound known as OB101 that can upregulate tonic inhibition in the brain and hopefully produce some clinical benefits. So this was a drug that was previously developed to try to help sleep indications, and now Ovid is taking it forward in the clinic for Angelman syndrome. So they've just completed phase 2 clinical trials, and they reported a significant improvement in a clinical global impressions of improvement, the CGI-I measurement, and now they're advancing to a phase 3 trial, which they're calling Neptune. And that's going to be commencing soon for individuals in ages 9 to 12 initially, and then later on for individuals ages 4 to 8 and ages 2 to 3. So their representatives from out here who should be able to speak to that in more detail. Obviously a really common problem in Angelman syndrome, affecting 90% of the individuals, is seizures. And there have been many reports of people trying different formulations of cannabidiol to try to treat seizures that cannot otherwise be controlled. So Dr. Ben Gu at the University of North Carolina did a very systematic study in mice to try to see if there was a strong rationale for using cannabidiol in Angelman syndrome. And the take-home message of this is that with the appropriate concentration of a very purified form of cannabidiol, it could really reduce seizure severity, with the caveat that it could also produce at high doses a mild sedative effect. But this does provide rationale that cannabidiol can be yet another anti-epileptic that's in the repertoire of anti-epileptics that might be used to treat seizures with Angelman syndrome. And once again, I would tell the parents to always work with your neurologist to figure out the best anti-epileptic approach. But you can always raise the possibility if other anti-epileptics are working, whether there's an opportunity to consider a purified form of cannabidiol. You can read about these findings because in an effort to get them out to the public quickly, you can now deposit them in a publicly available repository known as bioRxiv. And Dr. Bindu has published, or put his findings out there for the public to read if you'd like to read it in advance.
So, as you know, ASF is also thrilled to have a very large network of Angelman clinics. And I know you guys are thrilled because this gives you places hopefully closer to your homes to see specialists who treat Angelman syndrome. But they're also important vehicles for research, for researchers. And there's a lot of wonderful research that's going on in these clinics that help to figure out the best immediate treatments for your kids, and also to understand what's going on with your kids so that they can come back to researchers and say, find us something to fix that, and so on and so forth. So there are now 12 Angelman clinics worldwide in ASF's clinical network, everywhere from Boston down to Buenos Aires, Argentina. And so we're very excited to have ASF clinics go global. And they've been contributing to the research literature and important findings. One of the important things that the clinics are participating in are these natural history studies. And I've lumped them into one thing, but there's actually multiple different studies. First of all, there's the Global Angelman Registry in Australia, and that's got important parent-reported data. We encourage you to participate in that. But there's also studies funded. There was an older natural history study funded by the NIH that's yielded publications from Wen Han Tan's group. And there's also a new natural history study funded by the FDA. And what's really cool, in my opinion, is that there's collaboration between 3 pharmaceutical companies pre-competitively, meaning they work together to generate data that they can all share. That's supposed to identify clinical endpoints. And so this is, again, an attempt to get the natural history to understand what's going on with Angelman syndrome prior to therapeutic intervention. And so all of these studies are working together to give researchers a big picture of what's going on in your kids, so that we may be able to intervene in a meaningful way. There have been studies from UNC/RTI looking at anxiety in Angelman syndrome, and this is, I think, one of the most robust and up-to-date sources to understand anxiety in your kids that we're beginning to recognize more and more as a significant problem. And also this group from the clinic in Canada looking at behavioral approaches to assess memory, imitation, and motor performance in kids with Angelman syndrome. And these are important ways so we can understand how to measure your kids' progress in these areas, which will become increasingly important when clinical trials arrive. Additional studies, sleep has been studied in a collaboration with Ben's lab. This is out of Ben Philpott's lab, largely in the Angelman clinic at UNC. Myoclonus and Angelman syndrome out of the Mass General Hospital Clinic. And challenging behaviors in toddlers and preschoolers. We've just heard a wonderful talk from Bridget Thompson in the scientific seminar, talking about how she's able to measure behaviors and progress in very young children with neurodevelopmental disorders, including Angelman syndrome. And this work is encouraging for helping us understand what's going on in Angelman syndrome. So, with all of the clinical information, we've talked about basic science. Now it's going to be important to identify all of the individuals with Angelman syndrome. And there have been substantial bodies of work now trying to find ways for newborn screening. And what that means is that when babies are born, many states screen for a lot of different disorders on a newborn screening panel. And many of you may not have even known that your child was tested for various disorders. And it's an ultimate goal of ours to add Angelman syndrome to this list so that we can identify kids early and then to identify all kids with Angelman syndrome. So a paper was published earlier this year addressing one rapid and accurate assay to diagnose Angelman syndrome. But we also heard work from Dr. David Godler, and this was work that was we co-funded with the Foundation for Prader-Willi Syndrome Research and the Do 15Q Alliance, because his test ultimately can detect Prader-Willi, Angelman syndrome, and duplications of the same chromosomal region. And so working together in this case, we all win. And so ASF's dollars go farther, but Dr. David Godler talked about this rapid and sensitive test that is inexpensive enough to be included on a newborn screening assay. So what are, this is Ben's part,
So there are many challenges that are facing therapeutic development. So, how do we establish the optimal ages of intervention? And that's going to depend on the type of intervention. And how does this vary by phenotype? So what are the correct interventions for individuals who have uniparental disomy versus individuals who have deletion versus individuals who have mutation versus individuals who have mosaic? What brain regions do we need to target? How are we dealing with variability in the patient populations? And the patient population has variability in what their exact genetic mutation might be, but they also have other genes that are different as well. And how do we balance a desire to develop something that's curative, or as curative as it could possibly be, to the immediate needs of helping individuals with Angelman syndrome? So we really have a lot of discussions of how we should best prioritize our strategy treatments and what we should devote the most of our time to. How do we establish the safety of clinical trials? So we're trying to work closely with the pharmaceutical companies who are advancing towards clinical trials. And how do we not overburden the patient population? Because while it's exciting for all the families that we're getting close to many clinical trials, we're soon at the point where you might be asked to be in many clinical trials. So how do you decide which clinical trials to participate in or not participate in? But ultimately, we just have to remind ourselves that our goal is to improve the lives of individuals with Angelman syndrome. So every single thing that the Angelman Syndrome Foundation does has a singular goal in mind. Whether it's a basic research study performed in a mouse, whether it's a clinical study, whether it's a drug development program, the foundation is very careful to try to make sure that every dollar that's used is being used to improve the lives of individuals with Angelman syndrome. And we're going to end there, and hopefully we have time to answer the questions if you may have them. Thank you.
Thank you. Travis Blake, Atlanta, Georgia, so it's great to see you here. I'll follow your work. Thank you for coming. So one question I had, I hear a lot about UBE3A, and I wanted to see if anybody has any conversations about UPD.
Sorry, about UPD?
It's one of the diagnoses for
Yes,
HPD.
Yeah, we have.
yes. I just didn't know if I heard that right. So uniparental disomy, of course, means that you have 2 copies of chromosome 15 from your dad, and therefore you don't have one from mom. And so there are a lot of cool human studies that are trying to understand the differences in phenotypes between the different genotypes. So do kids with deletions have the same phenotypes? Do they look the same as the kids with uniparental disomy, and what are the subtle differences? And those are kind of important to understand, so we understand the treatment populations. My lab, for instance, has made iPS cells from individuals with uniparental disomy, so we can understand how different therapeutic approaches work in uniparental disomy cells versus deletion cells. And then, the mouse model that was talked about earlier with imprinting center defects is going to be somewhat similar to uniparental disomy as well. And those, again, all help us understand the differences between the different genotypes. And so it's very much on our radar, and there were talks that addressed that in the past couple of days.
If we can hear you, we can
Yeah,
repeat the
we can
question.
repeat it.
Just yell it out, we'll repeat it.
I don't have a question. I have a comment. And I think it's appropriate to put things in perspective because I came to tears listening to some of the things you're saying here a little while ago. I happen to have been to every ASF conference since 1989, and I recall in 1990-91, the ASF board of people who represented you debated, great discussion, trying to fund $10,000 so that we could replicate the first Angelman syndrome house at the University of California in Los Angeles. And we spent $10,000 on the hope that we could make an Angelman mouse that would enable research to move forward. So the therapeutics, that's a new word for everybody. And most of you probably don't really appreciate the work that's been done over about 30 years. But it is, I assure you, significant. And we're grateful to Ben and every collaborating scientist who's working on the potential of therapeutics as providing some relief for our children, young adults, and older folks who have Angelman syndrome. So I make that comment, congratulating you on addressing some issues which I assure you 30 years ago, none of us ever conceived would have taken place.
All right, thank you to Stormy and Ben.